Good morning, ladies and gentlemen. Welcome to the 2023 AGM for Island Pharmaceuticals Ltd. My name is Paul McLearman, Island's Executive Chair, and I will be chairing today's meeting. It's now 11:30 Eastern Time in Australia. We have a quorum of shareholders present. I declare the meeting open for business. I confirm that the meeting has been properly constituted. Today's meeting has been convened in accordance with the Corporations Act. That's the presentation deck. Is that one we want? Okay. I would like to introduce our board of directors who are present in person or by Zoom today. Dr. David Foster, Managing Director, and Dr. Anna Lavelle, Non-Executive Director.
Good morning.
Sorry, Anna. Our other non-executive directors, Dr. David Brooks and Mr. Albert Hansen, have been unable to attend the meeting as they're both in Northern Hemisphere time zones. In addition, I would like to introduce our Company Secretary, Mr. Peter Websey, and our Chief Financial Officer, Mr. Cameron Jones, who is also attending by Zoom. I'd also like to introduce Mr. Nicholas Benbow, Audit Partner from William Buck Audit, Victoria Proprietary Limited, who is attending by Zoom also. Our Investor Relations Consultant, Jane Lowe, also with us today, as some of her colleagues, and will facilitate any QA which comes through the Zoom channel. The agenda for today's meeting is as follows. First, there will be a chair's address. Then we will cover the formal business of the meeting, which will be followed by the Managing Director's address. I'll now present the chair's address. Ladies and gentlemen, the financial year 23 year was marked by both successes and challenges for Island. In December 2022, after many months of work, we submitted our investigational new drug application for our lead asset, ISLA 101. This was an important and critical milestone for Island. What we didn't expect based on our pre-IND meeting was a clinical hold from the US FDA and the need to conduct a small single ascending dose study to enable a progress onto our phase 2 clinical study. Today, we are at an important moment in Island's history. Just last week, we received approval from the Ethics Committee to commence the single ascending dose study. We are now commencing screening and recruiting patients for the study, which will help us when optimising our dosing regimen for the subsequent phase 2 PEACH study. We expect to move through the single ascending dose study rapidly, so after a period that we agree has been challenging, we feel that we are now on the road to quickly delivering data using the clinical stage asset that we have in ISLA 101. Given what's going on around the world, development of an effective antiviral drug to prevent or treat diseases like dengue fever, chikungunya, and Zika cannot come fast enough. Reports of mosquito-borne viruses continue to make headlines in 2023, underscoring a substantial unmet medical need for new treatments like ISLA 101. Dengue fever's range has now extended to more than 100 countries. Europe witnessed nearly as many domestically transmitted cases last year as it had in the previous 11 years combined. According to the latest WHO statistics, approximately half of the world's inhabitants are now vulnerable to dengue, with an estimated range of 100 to 400 million infections occurring each year. What keeps us up at night and what continues to fuel our commitment to propelling drug candidates such as ISLA 101 forward? Outside of the ISLA 101 program, the board approved a plan to explore acquisition or in-licensing of new drug candidates. Underpinning our work, we strengthened our team and advisors, and we're delighted to welcome Dr. Amy Paddock to Island's Pharmaceutical Board. In line with our commitment to strong regulatory engagement, we also welcomed senior regulatory consultant Bobby Drays. With her extensive experience in regulatory strategic management, Bobby has strengthened our in-house regulatory affairs expertise to support our ongoing communications with the FDA. In closing, I'd like to express my gratitude to our management team and board for their dedication and ongoing efforts throughout the financial year 2023. I would also like to extend my sincere thanks to you, our shareholders, for your continued support, especially given the challenges we've faced in the financial year 2023. With these challenges now resolved, we're excited to move ISLA 101 forward into the next phase of its clinical life and thereby raise the profile of the company with investors and potential partners. Thank you. Does anybody have any questions on the chair's address? We'll now move on to the formal business of the meeting. The notice of meeting was lodged with ASX on the 13th of October 2023. We will now take the notice of meeting and the resolutions to be considered at the meeting as read if there are no objections. Voting on all resolutions will be conducted by poll. For the purposes of the poll, I appoint Matthew Hunter of Atomic Group, the company's share registry, who have examined and prepared summaries of the proxy forms received, to act as returning officer and to conduct the poll. Shareholders, representatives and attorneys of shareholders and proxy holders who hold yellow voting cards are entitled to vote at this AGM on a poll. Shareholders who are not voting at this year's meeting would have been issued with a blue card. Please hold your blue or yellow card if you wish to ask a question. Proxies have been inspected and all those validly lodged have been accepted. Proxies have been received from 13 shareholders representing 19.4 million shares, being 23.9% of the company's issued capital. I will declare the proxies received on a resolution before putting the resolution to a vote. I would like to highlight that where undirected proxies have been given in favour of the chair, I will be voting these proxies in favour of all resolutions being put to the meeting today. We'll now move to financial statements and reports. The first item of business is to consider the financial statements and the reports of the directors and of the auditor for the year ended 30th of June 2023. The financial report and reports of the directors and the auditors are now laid before the meeting. There will be no vote on this item and it is a discussion item only. The company's auditor for the 2023 financial year, Nicholas Benbow of William Buck Audit, is present by Zoom to take questions relevant to the audit, to the conduct of the audit and the preparation and content of the independent auditor's report. Are there any questions or comments on the financial report or the reports of the directors and auditors? If you're attending the meeting online, please use the Q&A function in the Zoom ribbon to submit your question. Are there any questions or comments of the management of the company?
I don't believe there are any questions at this stage.
Are there any questions relevant to the conduct of the audit and the preparation and conduct of the auditor's report to be put to the auditor?
Not at this stage.
Thank you. We'll now proceed with the resolution set out in the notice of annual general meeting. Resolution 1 is set out in the notice of meeting is to consider as an ordinary resolution the adoption of the remuneration report. If you are present in the Zoom and wish to discuss this resolution or have a question, please raise your voting card. Alternatively, if you are attending the meeting online, please use the Q&A function in the Zoom ribbon to submit your questions. Jane, are there any questions?
No questions that I can see at this time.
The proxy votes received on this resolution are currently displayed on the screen. Voting on this resolution will be conducted by a poll to be held after all resolutions have been considered. We will now move to the next resolution. Resolution 2, as set out in the notice of meeting, is to consider as an ordinary resolution the re-election of Dr. David Brooks. If you are present in the room and wish to discuss this resolution or have a question, please raise your voting card. Alternatively, if you're attending the meeting online, please use the Q&A function in the Zoom ribbon to submit your question. Any questions?
No questions this time.
Thank you. The proxy votes received on this resolution are currently displayed on the screen. I don't think I've ever seen 100% resolution before. Voting for this resolution will be conducted by a poll to be held after resolutions have been considered. For the purposes of the poll, Resolution 3, as set out in the notice of meeting, is to consider as a special resolution requiring it to be passed by 70% of the votes cast, the approval of increased placement capacity. If you are present in the room and wish to discuss this resolution, please raise your voting card. Alternatively, if you're attending the meeting online, please use the Q&A function in the Zoom ribbon to submit your questions. Jane, are there any questions?
No questions at this time.
The proxy votes received on this resolution are currently displayed on the screen. Voting for this resolution will be conducted by a poll to be held after all resolutions have been considered. We will now move to the next resolution. Resolution 4, as set out in the notice of meeting, is to consider as a special resolution requiring 70% of votes cast the adoption of a new constitution. If you are present in the room and wish to discuss this resolution, please raise your voting card. Alternatively, if you are attending the meeting online, please use the Q&A function in the Zoom ribbon to submit your question. Any questions?
No questions.
Thank you. The proxy vote received on this resolution are currently displayed on the screen. Voting for this resolution will be conducted by a poll to be held after all resolutions have been considered. We will now move to the next resolution. Resolution 5, as set out in the notice of meeting, is to consider as an ordinary resolution the adoption of a new employee incentive plan. If you're present in the room and wish to discuss this resolution, please raise your voting card. Alternatively, if you are attending the meeting online, please use the function in the Zoom ribbon to submit your question. Any questions? Thank you. The proxy vote received on this resolution are currently displayed on the screen. Voting for this resolution will be conducted by a poll to be held after the final resolution has been considered. We will now move on to the final resolution. Resolution 6, as set out in the notice of meeting, is to consider as an ordinary resolution the appointment of auditor. If you're present in the room and wish to discuss this resolution, please raise your voting card. Alternatively, if you are attending the meeting online, please use the Q&A function in the Zoom ribbon to submit your question. Are there any questions? Thank you. The proxy votes received on this resolution are currently displayed on the screen. Voting for this resolution would be conducted by poll. Ladies and gentlemen, that concludes the resolutions to be voted on today. As noted, we are conducting a poll on all resolutions. For this purpose, and as I stated earlier, I appoint Matthew Hunter of Automic Registry Services to act as the returning officer. When you registered your attendance for this meeting, voting shareholders and proxy holders were given a yellow voting card. On this card, you will find a series of boxes for voting. For your vote to count, you must tick or mark either for or against or abstain box. As a proxy holder, you will have been provided with a report when you registered, which summarises how you have been directed to vote. When completing this card on the poll, you need only mark a box if you have discretionary votes. If you do not have discretionary votes, then you'll need only hand your card to the staff collecting the voting cards. When voting on the resolutions is complete, staff will collect your voting cards. I now ask that you complete your voting cards for all resolutions. There being no further questions, I now close the poll. The staff of Automic will now process the poll and the results will be declared on each resolution and released on the announcements platform of the ASX, published on the company's website once they are available. I'll now hand you over to our CEO, Dr. David Foster, to give his presentation, after which there will be opportunities for questions.
Thank you very much, Paul.
Thank you.
Thank you, everyone who is online attending the meeting. Thank you, everyone who is here attending the meeting. I've been in Australia for the last 2 weeks, having a lot of meetings, getting to see the clinical site where we're going to be conducting our upcoming clinical trial, meeting the CRO, meeting the IR department team face-to-face, and it's great to be face-to-face. I have to tell you, it's been a while since I've been back here, and I look forward to coming back soon. If we can go to the next slide. We're going to start at the beginning and give you all an overview of Island Pharmaceuticals and what we are, what we're doing, and why we're excited about it. At its heart, Island Pharmaceuticals is a drug repurposing company. We're focused on the rapid development of antiviral therapeutics for infectious diseases. A term you're going to hear throughout the presentation is repurposed drug, and we like the idea of repurposing molecules. By repurposing, I mean we're identifying a molecule that has a clinical history in one indication, and we're going to rely on the data basically from other people and use those data to move forward with a new indication where we have a proprietary angle. That's what we've done with ISLA 101, and it's a strategy that we like. Next slide, please. At a high level, just to give you a quick overview of Island Pharmaceuticals, we are moving forward with our lead molecule, ISLA 101, in dengue infections. Dengue is a huge problem. I'll show you some data here in a little bit, describing in more detail what the problem is with dengue. But on this slide, 400 million infections a year. That's about 1 million infections a day. It's a tremendous number of people who get infected with dengue every year, and there's nothing you can do about it. Vaccines are problematic. There's no small molecule that is effective as a therapeutic or a prophylactic. You have to treat symptoms, and it's a significant problem. As a result of that huge problem, that does create significant market opportunities. ISLA 101 has the potential to address dengue infections as well as other mosquito-borne viruses, both a prophylactic or preventative medicine, potentially as a therapeutic, and we're interested in exploring both. The reason we're very excited about this, ISLA 101 is a molecule that's well known. It's been around for 30 years. It was initially developed by Johnson & Johnson as a cancer therapeutic. It's been in thousands of people in the clinic. It's been in 40 plus clinical trials. It's never been approved for anything. It has a great safety profile. It's not an effective cancer drug. We have data, phenomenal preclinical data package demonstrating that ISLA 101 is effective against a variety of mosquito-borne viruses. This includes experiments in human cells, in animal models, different flavivirus infections in different animal models. It's a fantastic preclinical package. Where are we going now? We're moving forward with a single ascending dose study. This is, as Paul mentioned, was requested by the FDA. And we're moving forward. I'll give you some exciting updates as we get through the presentation. There's an interesting opportunity in the United States called a priority review voucher. We're currently eligible for a priority review voucher. Again, I will give you more information throughout the presentation, but that's a benefit that could accrue to us upon drug approval. As Paul mentioned, we are exploring opportunities for pipeline expansion, and we'll touch on that briefly. Next slide, please. The World Health Organization came out just a few months ago and made some really nice sound bites for us, quite frankly. About half the world's population is now at risk of dengue, with an estimated 100 to 400 million infections occurring every year. This is a staggering number of infections. And again, the prophylactics, the therapeutics, they're non-existent. We're excited about the opportunity we have with ISLA 101. Next slide, please. This is a map that I like to look at, and we update this about every 3 months to look at really the hotspots of dengue. Where is dengue popping up? The countries that are in red are endemic. The dots are signifying recent outbreaks. You can see Latin America has a significant problem. Basically, all of Latin America is endemic. It's a huge issue. Also, Asia has a huge problem. India has a huge problem. If you look at the United States, you can see there are some dots in the southeastern portion of the United States. This is in Florida, where they've had locally acquired dengue infections. If you look at Australia, you'll notice there's not a lot. It's interesting that Australia doesn't see a lot of dengue, although there are other mosquito-borne viruses to be concerned with. If we go to the next slide. This is a slide that I think is really important. This is an estimate of where dengue is expected to be endemic simply in the year 2050. This is 27 years down the road. As a result of global warming, if nothing is done to curtail global warming, we're expected to see dengue now endemic in the southeastern third of the United States, in the northern third or half of Australia, and now extending into the southern part of Europe. So today it's a huge problem. It's only expected to get worse. And again, there's really nothing you can do about it today. Next slide, please. Dengue fever, again, from the World Health Organization. Dengue fever will become a major threat in the southern US, southern Europe, and new parts of Africa in this decade. Next slide, please. Because there's such a problem, as I mentioned at the beginning, there are significant opportunities. We anticipate that ISLA 101 could be useful for travelers. Think like antimalarials. The antimalarial market is expected to approach $1 billion market opportunity within just a couple of years. So that's a significant opportunity. Likewise, we think the military may be very interested in what we're doing. In the United States, we actually have an agreement in place with the U.S. Army, a cooperative research and development agreement. I'll fill you in a little bit more on the details of that in a couple of slides, but the Army is a wonderful partner to have in the U.S., and we are very happy working closely with them. Because there's so many people in Latin America, in Asia, we believe that there's an opportunity for ISLA 101 to be a part of government stockpile programs. And we have been speaking with people in these different jurisdictions about how to navigate getting to the government to support such a thing. Again, I've mentioned at the beginning the priority review voucher program, for which we are currently eligible. This is a program created by the FDA in the United States to incentivize companies to develop drugs for underserved diseases. And the way it works is if you get your drug first approved in an indication on a list of diseases that the FDA has established, then you get a priority review voucher. Getting priority review at the FDA is a phenomenal thing because it expedites your review period at the FDA, allowing you to bring a drug to market sooner than you might otherwise get. Big pharma companies like Merck, like Pfizer, they love the opportunity to get priority review to bring their next GLP-1 agonist to the market. The beautiful thing with these vouchers If we would get one upon approval of ISLA 101, as you can sell these vouchers, they typically sell for about $100 million US. So you continue to keep your program, you commercialize your program, but you get this ticket or this voucher that you can transact. And Big Pharma likes to buy these up, again, for around 100 million US. We like the opportunity. And just to be clear, it would be available to us at approval. So we still have some time to go, but we're excited by the possibility. Next slide, please. So what is ISLA 101? I teased at the beginning, ISLA 101 is a fascinating molecule. We really like working in it because of the robust clinical history. It's been in thousands of people in the clinic. It's gone through phase 1, phase 2, phase 3 clinical trials around the globe. It's never been approved for anything. That means there's a huge database of data describing the safety of the molecule, and we're relying on all of that, as well as some information we received from the National Cancer Institute. We're able to reference an investigational new drug application that the National Cancer Institute holds. This is helping us expedite our way into clinical trials, and we're relying on all of this information. ISLA 101 has activity against all 4 strains of dengue, which is really important. The 4 strains of dengue create a huge problem for vaccine development. In order to have an effective vaccine, it needs to be effective against all 4 strains of dengue, and it needs to provide around 90% protection against all 4 strains. That's a very high bar. It's a significant problem, and it's something we anticipated when we started this company, that the better approach, in our view, was to have a small molecule that could be a prophylactic as well as a therapeutic. And that's what we have in ISLA 101 and what we're exploring in the clinic. The preclinical data package we have, again, is fantastic. Data that ISLA 101 is effective against all 4 strains of dengue, as well as Zika, West Nile, yellow fever. These are all flaviviruses. This is the only pan-antiflaviviral molecule that I'm aware of. And I'm excited by the possibility that would provide for us as we move this forward to approval. The preclinical data package we have, again, we have data in freshly isolated human cells demonstrating that ISLA 101 prevents the infection of cells in humans. These are human cells in culture. We also have preclinical data in animal models, 2 different animal models generated by 2 different universities. This is really important. It's not just one university. Monash University did work in dengue animal models, which I'll show you some data from this shortly. Likewise, a group at Harvard, at the same time that Monash was looking at dengue and ISLA 101, the group at Harvard also performed animal studies demonstrating that ISLA 101 protected against infection in a dengue model, as well as a Zika model. So fantastic data. We're very excited to move this forward. Next slide, please. This is the only data slide that I'm going to show you on science. What we're looking at here is a survival curve, where along the y-axis, we have percent survival from 0 up to 100%. Along the x-axis, we have time after infection. This is an infection model of mice that is a lethal model, so the mice will all die. And this is shown, if you look at the red trace here, 5 days following infection, there's 0% survival of these animals. They're all dead. However, if at the time of infection, you start dosing with 1 dose a day of ISLA 101, about 20% of the mice survive. This is fantastic. If you dose with 2 doses a day, it gets even better. 70% of the mice survive in this otherwise lethal infection model. It's actually a model of dengue hemorrhagic fever, which is aggressive as you can possibly get. As a scientist and a drug developer, when I see dose responses like this and you see an increasing dose proportionally results in increased benefit, this makes me very excited. So in view of the robust clinical history we have with ISLA 101, the phenomenal data package we have with ISLA 101, we're excited to get into the clinic in humans, which we're headed towards. Next slide, please. The progress we've had over the last year, Paul has given you some highlights already. Last December, we submitted our IND to the FDA for our phase 2 clinical trial. I'll refer to this going forward as the PEACH clinical trial. It's a prophylactic challenge study, which I'll provide some more details shortly. In January, February of 23, to our surprise, we received feedback from the FDA where we were put on clinical hold. The FDA asked questions really about our manufacturing and wanted to ensure that the drug we had manufactured behaved in the same fashion as other people who have used ISLA 101 formulations. We quickly responded to the FDA. The clinical hold has been lifted, but we do need to move forward with this dose escalation study, which we're moving forward with now. Along the way, some beneficial news that we received, really very happy news. in collaboration with our partners at SUNY Upstate, where we're going to be doing our phase 2 clinical trial, SUNY received a $1.3 million grant to support studies out of our clinical program. There will be studies that we don't have to pay for, but data that we will own, and they'll be doing high throughput genomic sequencing, deep sequencing, genomic analysis, looking for biomarkers associated with a dengue infection. It's going to be a fantastic set of data, again, that we'll all own. Next slide, please. Moving through towards the single ascending dose study, it's been a very fast and active period recently. I don't want to undersell the importance of this first box that we checked, but we have manufactured the material. The material has been labeled, it's been packaged, it's been shipped to Australia, it's been imported into Australia. Last week, I had the great opportunity to visit our clinical site and see ISLA 101 sitting on a shelf, waiting to be used in the clinical trial. This is a fantastic day for me. Frankly, I really wanted to take the picture, but I decided not to because there were other companies there who are also doing clinical trials at the site. We've signed the clinical site, which is Scientia. They are in Sydney, a fantastic site. I had the great pleasure to do a site visit last week and was very impressed with the situation they have going on there. Beyond Drug Development is our CRO. We've engaged them. I've had the good opportunity over the last 2 weeks to meet with them face to face. So it's been a very productive time here in Australia. And then the day that I arrived last Monday, we obtained ethics approval, giving us the green light to move forward in the clinical trial. So the timing was great. I arrived, we had ethics approval, drug is on site. Then we found out last week that we had been approved to start screening. They are screening patients. Now they're screening healthy individuals to enroll in our clinical trial. And I spoke with the site this morning. They are actively screening people. We have people who are now having blood samples taken and doing the blood work before we can enroll them, but it's moving along very nicely, which we're very excited about. So we expect to be dosing by the end of this month with the first cohort, and I'll tell you about the clinical trial design. It's 4 cohorts. Looking at increasing doses of ISLA 101. We'll expect to wrap up dosing by early 2024 with data coming out soon after that. If we could go on to the next slide, please, James. The single ascending dose study, I won't go through all of this in great detail. I think it's on our website. It's healthy individuals that will be enrolled. It is 4 cohorts of 8 healthy individuals. And again, the first cohort comes in, they get 1 dose. Then we take blood samples over the course of the next few days. And the question that we want to ask is, 1, is it safe? We feel like it should be safe in view of the enormous body of literature that we have available at our disposal right now. The other question we want to ask is, if I give a particular dose to this individual, what's the resulting blood concentration? That's really important for us as we look to our phase 2 clinical trial coming down the road, because we want to ensure that we dose very carefully to hit the dose that will be effective against a dengue infection. So after the first cohort here gets their first dose, the second cohort will get an increased dose. The third cohort will also get an increased dose. The first 3 cohorts, these are all people taking this drug without breakfast. It's under fasted conditions. The third cohort actually is going to come back for another treatment, and we will dose them after we give them breakfast. We want to see what effect food has on the blood concentration at that higher dose. I think that's it. Let's move on to the next slide, please. Our partners, I've already spoken with them. They're fantastic. Scientia here in Sydney. And then Beyond Drug Development is our CRO. They're based in Brisbane. I had a chance to meet them in Adelaide last week. Next slide, please. One of the other things, before I move on, about the dose escalation study that we're going to be doing in Australia, because we're using Scientia, because we're using Beyond Drug Development in Australia, 100% of the spend associated with this clinical trial is eligible for the R&D tax credit. So we're very excited about that. The ISLA 101 development timelines, again, we expect to have data coming out of this dose escalation study early calendar year 2024. We'll likely seek a conversation with the FDA to discuss with them the results from that study, see if there's ways we can maximize the data we move forward with the PEACH clinical trial, the challenge study that I'll tell you about. We may need to amend the protocol based on the dosing data that we get out of the dose escalation study, but then we want to move forward with the challenge study as quickly as we possibly can. Again, we have the protocol already written. We have partners already lined up. The site is ready to go. We essentially need to turn things back on and move forward with this phase 2 study. Next slide, please. The phase 2 clinical trial, I'm excited to give you an overview of this. It's a challenge study. And by challenge study, I mean we're going to enroll healthy individuals and we're going to infect them with an attenuated or a weakened strain of the dengue virus. People have been doing challenge studies for years. We saw a lot of challenge studies occur through the course of COVID. And the experts and the regulators are recognizing it is a very efficient way to look at a small number of people in a very controlled setting and obtain huge amounts of data about the way a drug may interact with a particular virus. The dengue virus is very interesting, and it's one of the benefits of running clinical trials in infectious diseases because it's lightning fast. From the time you infect one of these individuals with dengue, the dengue viral load peaks at about 7 days. By day 14, it's all gone. And so what we're able to do with these individuals, we're going to do a prevention study. So we will enroll these individuals, pre-treat them with ISLA 101, then infect them with the attenuated dengue virus, continue to treat them for the next 20 days. But the goal here is to see if we can suppress the infection, reduce the duration of infection, or suppress the symptoms associated with a dengue infection. One of the benefits of this study is we're doing it in collaboration with the U.S. Army. The U.S. Army developed the virus strain that we're going to be using in the clinical trial. We have a cooperative research and development agreement in place with them, under which they're giving us the virus. They're also giving us the data that they have obtained after a phase 1 study that they ran. They ran a study just looking at the virus, the effect it had on the people who were infected with it. And I can tell you, people got a little bit achy. They had mild flu-like symptoms. Some people developed a rash. Most people develop a rash. Some people developed a fever. Some people developed a headache. But the patients, the individuals in the study, they take a diary and keep track of all the symptoms throughout the duration of the trial. They also come to the hospital and they take blood samples every day. So you have the opportunity to measure the viral load in the samples of the individuals who have been infected. You can measure antibody responses against these individuals. You can measure biomarkers associated with infection. It's incredibly powerful. Because of the way this is set up, the number of individuals we use is very small. It's only 16 people. It's 4 cohorts of 4 individuals, each with the opportunity to start with a dose of ISLA 101, and we'll either dose escalate in the second cohort, or we'll dose de-escalate depending upon the results from that first cohort. One of the great things about this and the dose escalation study is it's going to result in significant news flow over the course of the next year. We'll be talking about advancing through the different cohorts in the dose escalation study, as well as in this the PEACH clinical trial here. It's going to be a very exciting time for us. We can move on to the next slide, please. Paul mentioned at the beginning that we are also exploring opportunities for pipeline expansion. And we are looking at all of these opportunities through the same filter that we looked at ISLA 101. Is it a repurposed opportunity? Is it a small molecule program? Might it be eligible for a priority review voucher? That's something that would be important to us. Is there an opportunity for non-dilutive funding? We think that's really important. We're big fans of non-dilutive funding opportunities. We're talking to people. We're looking at a variety of things. Obviously, no decision's been made, but it's something that we do explore. Next slide, please. To talk about our fantastic team, I'm incredibly excited and grateful for the opportunity to work with this board. Dr. Paul McClayman, fantastic chair, executive chair, very happy for the opportunity to work with him. Dr. David Brooks, fantastic, phenomenal experience, very valuable at the board level. Mr. Albert Hansen, one of our U.S. directors who's been with the company for many years, significant experience in the industry and brings a lot of experience to board discussions. Dr. Anna Lavelle is fantastic, phenomenal, engaging at the board meetings, spot on with her issue spotting, but then nonetheless, a lot of fun to be with. So thank you all for the opportunity to work with you on the board. Next slide, please. Paul mentioned at the beginning, we have brought Bobby Drays into the fold. Bobby's a regulatory consultant. we're going through clinical trials, there's many factors that you have to consider, from drug manufacturing to how do you set up your clinical trials to how do you navigate the regulatory environment. Having people on our team to work with our service providers has been invaluable. Bobby and Teresa both have been phenomenal. They keep check on the people that we're working with. We're moving along great as a result of having them on board and working closely with us. Next slide, please. Finally, our scientific advisors, phenomenally gifted, talented virologists, drug developers. Dr. Lee Farrell has years of experience in drug development, has drug approvals under his belt. Dr. Stephen Thomas is at SUNY Upstate in Syracuse, which is where we're going to be running our PEACH clinical trial. Dr. Thomas is kind of the gatekeeper of the PEACH study, and a world-class virologist, drug developer. He was actually recently in charge of Pfizer's phase 3 COVID vaccine study. So there's a decent chance you've seen him interviewed on the news. He's all over the place. Dr. Amy Paddock, I'm so happy that we've had the chance to bring her onto the scientific advisory board. She has years of experience in antiviral drug development, including experience in challenge studies. So she has a phenomenal background. Next slide, please. Completed milestones and upcoming milestones. It's going to be a phenomenally exciting year coming forward. Looking here in the middle, we're continuing to get patents issued. We recently had US and Australian patents issued. These are out of the portfolio that we've licensed from Monash University. As we very proudly announced at the beginning of this, we recently obtained ethics approval, HREC approval to initiate our dose escalation study. We've started screening that study already, and we'll be able to tell you about when we dose that first individual, and I'm excited about that. As I mentioned, we will just tell you when we're moving through to the different cohorts as we go through the dose escalation study. By early 2024, we should have the results of that study, and we'll be lining up to move into the PEACH clinical trial. Next slide, please. That might be it. Are there any questions in the room or from the Zoom?
We don't have any questions online. Any questions online?
There's no questions. I'll turn it back to the chair, Paul.
Thanks, David.
Thank you.
Thank you, David. If any shareholder in attendance has any general questions related to the conduct of the meeting, please raise your hands. For those attending via Zoom, please submit your questions using the Q&A function at the bottom of your screen. I'll give you a few seconds to type. Nothing coming through.
No.
Okay.
Thank you, Jane. Thank you, ladies and gentlemen. That now concludes the meeting. I'd like to thank all shareholders for their attendance today, and I declare the meeting closed. Thanks, everybody online.
Thanks, everyone.